One of the major motivations for launching the meaningful modeling of epidemiological data, or MMED, was to try and show students in the area that before you start getting involved in complicated mathematics, which may not even be necessary, what you need to think about when you approach a problem in the world of epidemiology is getting the biology or the medicine right, and then looking very, very closely at the data before you go anywhere else. And in particular, if you're presented with a set of data on some biological or medical or biomedical problem, you want to have three goals in mind to start with. First of all, when you get the data, the first thing you want to do is to look at the data. Just look at it, see if you can see any patterns, see what they might be telling you. And having done that, and particularly if you've been asked to solve some sort of problem, you formulate in your mind some manner of hypothesis, some sort of idea about what might be going on with this problem, where it's going to go. And having done that, you then set about trying to test the theories or the hypothesis that you've put forward. And keep in the back of your mind what Colin said, as is written down at the bottom of the slide. The purpose of models is not to fit the data so much as to sharpen the questions. So we will be trying to figure out what's going on with these data so that we can actually get better questions that we can ask of the data. And if you think that this is something that we just sort of sucked out of our thumb, let's actually look at a very serious scientist, and they don't come much more serious than Albert Einstein. And he said essentially the same kind of thing. The supreme goal of all theory is to make the irreducible basic elements as simple and as few as possible without having to surrender the adequate representation of a single datum of experience. Now, let's break that down. And the first half of what he's saying is exactly what I've been saying in the first couple of slides, is don't get involved in something massively complicated right at the outset. It may eventually be required of you to do that, but what you should be looking at first is some simple solution to go forward with. And that should involve, if at all, just as few parameters in your modeling as possible. But at the same time, when you look at your data and think about your data, you must make sure that you adequately represent everything that you've actually got in that data set. And this leads us to three basically good rules of modeling. The first is that you stay as close to the data as you can. So you get a set of data, and basically you're going to be trying to explain what's going on. What you have to do is think first, last, and always about the data. How can we make sure that the model that we've got actually gives us a decent representation of the data? And then, of course, you must put in as much biology, for which read biology or medicine, whatever. You must make sure that you have got a model which makes biological and or medical sense. There's no point in having a very complicated mathematical equation if actually you look at it and you realize that it doesn't actually make any biological sense. It doesn't matter how clever that equation is, it will be meaningless. And that leads to the final thing, which is basically keep things simple. Keep things as simple as you can. Then it's particularly easy to see when you've made a mistake. Now, let's just take we're going to look at some examples of where people have had to wrestle with problems, biological, medical problems in the past. And we're going back right now to the middle of the 19th century, and to a man whom I'd never heard of before I was introduced to this topic, and most of you probably won't either. But his name was Ignaz Semmelweis, and he was a junior doctor in Austria, in fact, in the Vienna General Hospital. And he was greatly involved in looking at maternal mortality. which, as we're about to see, was a major problem in Austria and indeed the whole of the world, and particularly Western Europe at that time. So let's have a look at this, and we will look at rates of maternal mortality. Now, somebody gives you these data. If you don't actually look at it too closely, you think, oh, well, there is a certain amount of mortality there, but it's only 0.05 or something like that. Until you think, what does that mean? What does this mean? What it's basically saying is that you've got 0.05, so that's 5 in 100, or 50 women out of 1,000 died of puerperal fever while they were giving birth. Now, you don't have to know too much about the modern world to know that that would be completely unacceptable in anywhere in the world, but particularly in a place like Austria and in Western Europe. And yet in those days it was regarded as almost, it was the way things were. People thought about, well, childbirth is a very dangerous time for the baby and also for the mother. So people die, that's it. So what it basically is saying is that 1 in 15 mothers were dying of puerperal fever in childbirth. Now, the people in Austria and doctors at that time had no idea what was causing this fever. They just knew that people were dying. Puer actually is Latin for a child. So and there we are. So basically, it's just saying the mother gets a fever and she dies. And it's of course it's very sad, but that is the way of the things. And if you look at it, it's basically sitting around the same sort of level, 1 in 15. 50 in 1,000 mothers dying, and that's what it is. So, what Semmelweis actually looked at these data, and he discovered actually that there's something strange. Now, again, this is not involving any mathematics. He just looked at the proportion of mothers who are dying amongst two groups of women. The women shown with the blue dots and lines were those mothers where the attending person looking after the childbirth was actually a doctor, a junior doctor, even a student doctor. Okay, and then in the red dots and lines basically are showing those mothers where the childbirth was overseen not by a doctor at all, but only a midwife. Now I say only a midwife, okay. What I'm saying is the midwife, of course, does not have a medical degree. So she, by definition, has had less medical training than the junior doctors. And yet what you can see is that after about 1838, the proportion of women dying when they're being looked after by a junior doctor is markedly higher than amongst the women who are being looked after by a midwife. Now, this is the kind of thing that I'm sort of saying. We don't need a mathematical model to actually see what's going on here. We can see there is something very, very wrong, and that's precisely what Semmelweis said. How can it possibly be that these women who don't have any medical training have much, much lower rates of death amongst the women that they're looking after than the junior doctors? Now, one of the things that Semmelweis knew is that the medical students, when they were delivering babies, they could very easily have been doing an autopsy, i.e. they could have been cutting up a dead person to find out what that dead person died of, straight before they came into the delivery ward. And in those days, it was kind of a badge of honor. The more blood you had on your tunic, the more work you'd been doing. And so, this is kind of part of the thing. It's an appalling thing to think, but it was basically what was going on. So Semmelweis looked at this and he wondered what on earth could it be that is actually causing this huge difference between these situations. And he was thinking about that when one of his colleagues, an unfortunate man by the name of Jakob Kolechka, cut himself with a scalpel, one of the student scalpels, while performing a post-mortem, and he died with a pathology which is similar to that of the mother's. That's to say, he looked as if he had died of puerperal fever. Well, of course, he wasn't pregnant, of course, being a man, but he died of something which looked like puerperal fever, and this got Semmelweis to thinking, what on earth might be going on here? And so he decided that basically what the problem was, that somehow or another there was infection being spread by the doctors, which was not being spread by the midwives. And so what he did was he managed to, he was a junior doctor, by the way, so he was sort of going a bit out of line. He made the medical students wash their hands in a really strong solution, chlorinated lime, which is going to kill any germs that they might have on their hands before they went into the maternity wards. And what he found was, hallelujah, that the mortality, as you can see now with the blue lines and dots, basically dropped until it was the same sort of level as was seen amongst the women where their babies were delivered by midwives. Okay, so what Semmelweis has done, he hasn't done any mathematics. We'll talk about this in just a minute. What he's basically done is he's looked at the biological medical problem, and then he's looked at the data, and then he's got some sort of hypothesis about what might be going on, and he thinks maybe it's to do with washing hands. Now, we find this very difficult to believe in the 21st century, that people, in what seems like relatively modern times, didn't understand the importance of washing hands. But what one needs to remember, of course, in part, is that they had no idea about bacteria. They did not know how these sort of things might be transmitted. Okay, they just knew that somebody was getting a fever, and had no idea where that fever actually came from. Nonetheless, what Semmelweis did was to say, well, let's suppose that it's actually come to do that. Let's give that a try. So he forms a hypothesis, and then he takes some action. So he does an intervention where he gets these young student doctors to wash their hands, and hallelujah, to see if it works like a charm. Okay, this is really interesting. Now, before we leave this slide, let's just think a little bit about it. Okay, so what I haven't mentioned is that each one of these dots, so the way that you get the dots are giving you the estimated maternal mortality, and that's just a simple ratio of the number of women who died out of the total number giving birth in that particular year. Okay, but then going through those, each one of those dots is a vertical line, which gives you what you will know about. I would think it's a standard error, or actually in this case, it's a confidence interval, which basically says I don't want to get too much into the details of this, but it's essentially saying how confident can we be that this estimate that we've got of a maternal mortality is actually at the level that we put it on that graph. And what these quite small bounds are showing us is that we can be pretty sure, for instance, that where there is a big difference between the red curve and the blue curve, say between 1840 and 1847, that the difference between the red and blue is so great that it is not happening due to chance. There's something funny going on, which is basically what Semmelweis discovered. And when he made the young doctors wash their hands, then, as we say, hallelujah, they stopped being the cause puerperal fever. They stopped being associated with the high rates of puerperal fever which had been seen in the women that they were attending to before. So this is where mathematics, but it's very simple mathematics. It's mathematics, statistical mathematics, is actually telling. But we still don't have anything, and we're not even going to see a mathematical model. But we have, Semmelweis has solved an epidemiological problem by just going through the very simple processes that we've talked about before. Now, you might think, so this man has now done something really stupendous, and the people who run the hospital and perhaps the Ministry of Health or its equivalent in Austria in the middle of the 19th century would give a medal or give him a promotion or whatever, make him head of the hospital. But in fact, no, that's not what happened. He was actually fired in 1849, basically because he was criticizing his superiors. And you can imagine, I don't know exactly why it was that what he was doing, criticizing his superiors, but you can tell that if in fact he was able to see that there were things going wrong relative to the matter of hand washing, he might have seen other things going wrong in the hospital, and he was shooting his mouth off, and so they fired him. And so the poor man actually was fired, and you can see that in fact only 16 years later he died of septicemia, by which time he was only somewhere in the region, I think he probably only in his late 40s, in an insane asylum. The only good thing that comes out of the whole thing is that if you look at the graphs after 1849 when he was fired, in fact things are still pretty good, relatively speaking. They're still very much higher levels of maternal mortality than would be acceptable in the modern world, very, very much higher, but the doctors are not doing any worse on average than the midwives. So there we are. So we can do something to sort of find out, exactly what's the ratio. So again, we can actually do some very simple mathematics by looking at what's known as the odds ratio. That's to say, what's the probability of a woman dying given that she gave birth under the direction of a doctor, as a ratio of the same giving birth under the care of a midwife. Okay, so if you've got on the y-axis there, if you've got a value of 5, what it's saying, which is what happened around about 1847, that's saying between 5 and 6. What's that saying is that you're 5 times or 6 times as likely in 1847 if you're a mother giving birth. If you're seen to by a doctor, you're around about 5 to 6 times as likely to die of puerperal fever than if you were looked after by a midwife. And you can see over that whole period between 1840 and 1847, it was very, very bad. They were very, very high. The odds ratios are extremely high. Thereafter, they're sitting around about 1. And if you look at the confidence intervals, you see in general, or a lot of them are either very close to 1, or they even overlap. Okay, so this is ways that we can actually use mathematics in a simple way to see what's actually going on. Okay, so that is an example, puerperal fever, where somebody came in and said, okay, I can see what's going on here, and then actually formed a hypothesis of what was going on and tested it, and in fact it made a major difference. And of course it basically, ultimately, that was taken on by the medical profession, and they got much more stringent about matters of hygiene thereafter. So now let's look at something, a quite different disease, and we're going to look at cholera. Now, cholera was a major killer in the 19th century, and even in times, and can be even to this very day. But it was a completely disastrous disease in Western Europe, and where we're going to be looking at it in Great Britain, and in particular in England and more particularly even in London. And nobody quite knew. They knew that people died of this ghastly disease, but they had no idea about what was causing it. As I say, this is before people had any idea about bacteria or germs, if you like, which are causing a problem, and they certainly had no idea about where they were coming from. But it didn't stop people from having ideas. And William Farr in 1851 basically said, yeah, I can figure out what's going on here. He called it the miasma theory. And he thought basically it was because cholera was the result of people breathing polluted air. Now, this is not a crazy idea. This is London he's talking about. And in the 1850s, people were pouring their night soil, as they call it, i.e. the buckets full of their feces out of the window into the street over the night soil, it was called. There was very little by way of decent sanitation with water and one thing or another. So it's quite possible that in the air there are nasty things that you're going to breathe in, which are going to give you cholera. And he basically said that the amount of organic matter and its distribution will bear a resemblance to the law regulating the mortality from cholera at the various elevations, by which he meant that there is an effect of the altitude at which you live, which will impact what's going on with cholera. And he didn't just suck this out of his thumb. He looked at the rates at which cholera occurred in the region of London and going out of London and going up further north to areas where you've got higher altitude. And what he finds is, hallelujah, that actually if you get up to over here at above more than 300 meters above sea level, there's virtually no cholera at all. But as you go from right to left on this graph and you get down to 100 meters, then it starts getting a little bit higher. And thereafter, it really rises quite quickly as you go down towards sea level. And once you get up to sea levels, you get to a situation where you've got a cholera mortality of 100 per 100,000. Now, this is a lot. And moreover, if you look at the fit to this these data, you would say, yeah, this guy has got the right idea. And there are a number of things that can be said about this. And one of them is the thing that I told you about it, is that we warned about, is that the whole business of mathematical modeling. You don't need to get involved in complicated mathematical modeling. And the other thing that I did not say is that you need to be very careful not to be seduced by clever mathematics, which seems to give you the right answer. This seems like it's got to be true. If you look at the data, you fit the data. I've said, look at the data and come up with a hypothesis. He came up with a hypothesis. He tested the hypothesis, and it seemed to work really well. But there is a very big but here, and it is one thing that we need to think about the whole time when we're looking at playing with data and fitting data, and that is what we've seen here is a correlation. There's a very high correlation between the rates at which you're seeing cholera and cholera deaths in particular, and the height above sea level. But correlation is not the same as causation. You cannot make the conclusion from this graph that it's actually the mortality is not actually a function of the being above sea level. Okay, so in 1854, another resident of London says, and who I do not know exactly why it was that how he came to the idea that it wasn't something in the air, but he decided that it was much more likely to be a waterborne disease. And again, he looked at this and he formed a hypothesis. And then what he did was he started generating data, and he looked at things in very much more detail within the area of London. So he's not actually looking at changes in altitude. He's just looking at the streets of London, and here we are. These are, if you know anything about the UK, you've probably heard of these streets: Oxford Street, Regent Street. These, by the way, in the modern age, are very, very fancy areas, very high-class areas. One thing or another may not have been. They might have been. Those streets themselves might have been quite sort of upper class, but right next door to them, you've got a workhouse there, for instance, and you've got clearly areas where there are not perhaps such salubrious housing. Okay, so what Snow does is he generates data. He looks to data. So if we look at these red spots around the place, the more red spots you see and the more red you see, basically that is where all the cases of cholera are occurring. He just records these, and he sees that there's quite a big clump around this, where we see this workhouse in the region of Broad Street, and he wonders, I wonder what on earth is going on here. And then what he also does is he looks at the water pumps. Now he's got this idea that it's a waterborne disease. Well, somebody looks at this and says, well, I don't know what he's talking about really, because if you look along Oxford Street, there are water pumps there. There's no cholera whatsoever. There's no cholera. Most of these pumps are not associated with in their immediate vicinity, there are not too many cholera cases. But there was one, which was right in the center here, which was Broad Street, where there were an awful lot of cholera cases around and about Broad Street. And basically to sort of take his hypothesis further, what he does is he goes and he actually he essentially removed the handle from the pump it's a very famous case of the Broad Street water pump, so that people could not get water from that pump. So they had to go to some of these other pumps. And what happened was that, in fact, when he did that, the whole cholera epidemic collapsed completely. But just like poor old Semmelweis, actually, whereas he quite clearly showed what was going on, he was not widely appreciated. In fact, he was not appreciated at all. So Lancet, which is one of the world's most famous medical journals, and certainly the most famous British one, wrote in 1858 when he died, they said quite simply, this well-known physician died at noon on the 16th instant at his house in Sackville Street from an attack of apoplexy. And they said, all they had to say about him was his research on chloroform and other anesthetics were appreciated by the profession. Not a single mention of cholera, not a single mention of the fact that he was almost solely responsible for showing what was going on with cholera, and that it was actually a waterborne disease. So there you are. That then what actually happened, it was shortly after this that people actually started to get the idea that there are causative agents for these diseases. It would have been the same for puerperal fever. And what Robert Koch did in 1884, he became famous for his identification of the cholera bacillus, among other things. And he's acknowledged as the discoverer of the cholera organism. Okay, so this is the actual thing which is causing the problem, and it is actually in the water. Drink that water and you're going to get with the Vibrio cholerae in it, and you're going to stand a very high chance of getting cholera. Okay. And in 1854, Filippo Pacini basically found, he discovered a bacillus which he called a Vibrio, and described the organism in its relation to cholera. And that was recognized only in 1965. Now, it took from the middle of the 19th century until the year 2014 for Lancet to put out this rather lame retraction, frankly. And what they had to say is that Lancet wishes to correct, after an unduly prolonged period, they don't say that it's well over 100 years, and an impression that it failed to recognize Dr. Snow's remarkable achievements in the field of epidemiology and in deducing the mode of transmission of cholera. Comments in 1855, such as, "In riding his hobby very hard, he has fallen down through a gully hole and has never since been able to get out again. And has he any facts to show in proof? No." And they say, rather lamely, they were perhaps overly negative in tone. Well, draw your own conclusions. Okay, so let's now just fast forward to the 20th century and look at some historical data. And hopefully we will. Okay, so what you've got here are a set of graphs, and they need a little bit of explanation. But essentially, what you've got is a set of 5 graphs, and each one of them has got an x-axis, which is the time axis going from 1850 to the year 2000. And it's basically all-cause mortality amongst men, or should strictly speaking say males, because it's obviously babies and young boys and young men. But in England and Wales between 1838 and 2002 is basically what it's showing. And it is particularly highlighting deaths from various things. One, cholera, and then flu, and also deaths due to war, and finally due to smoking. So those are the vertical arrows you'll see. And so what you do see, and we may feel that the modern era is pretty bad, but if we actually look at what has happened to the deaths per 1,000 per year, you see they are falling really quite remarkably between 1850 and 2000. Hence or otherwise, of course, and this is happening around the world, hence or otherwise, we have got a massive increase in population, despite the fact that the birth rate is also declining. But if we look at these graphs, what we do see is that the deaths due to cholera, flu, war, etc., they're not happening. There are not peaks in these diseases all the time. It's not uniformly high. So, for instance, and by the way, it can be quite different for people of different men of different ages, males of different ages. So, well, if we look at cholera, so there was not very little death. There were very few deaths amongst the males 0 to 4 in 1850s. But if you actually look, and then not so many between 5 and 14. Between the ages of 15 and 34 was higher and higher yet thereafter. In the older age categories, you don't actually see a spike at all. If you look at influenza, what we see is, again, no deaths amongst the males between 0 and 4, some between the ages of 5 and 14, but then this absolutely horrendous spike. And in fact, it comes at the end of 1918, when men between the ages of 15 to 34, there's just this vast number of deaths. And it's actually particularly savage to look at it. If you look at the deaths as a consequence of war, what you see is that you have got severe deaths in the First World War, but they actually really pale in significance when you compare it with the number of people dying from flu. Okay. So you should look at these and just sort of think about what they're actually showing, because it's a very interesting situation. Just to emphasize quite how bizarre the influenza or flu epidemic was in 1918 to 1919. Just look at this graph and look at the x-axis, which basically, we're not talking about years now, we're just talking about months. And there was a blip of flu in July of 1918, which was leading up towards the end of the First World War. The First World War actually finished at 11 o'clock on the 11th of November of 1918, and by that time there was a serious spike in the proportion of people dying in the United Kingdom. So these are deaths per thousand. So you've got up to 25 deaths per thousand of just a single disease. And what is quite remarkable about it is that it goes through this amazing spike starting in October, and it's up to 25 per thousand by the end of November and into sort of Christmas, and then dies down to almost nothing in January. There's another blip in February, March, and then it vanishes. And in that time, nobody knows how many people died in the world, but it could have been 20, it could have been 50 million. Anyway, it was an order of magnitude more than all of the men killed in the trenches in the First World War, which was the most barbaric war probably fought ever, and certainly the most barbaric one fought in Europe. So it's quite interesting, of course, because if you fast forward from 1918 to 2018, what happened in 2018 was that there was a sudden panic when we had COVID, and it is almost certain that in the back of everybody's mind was that we had a situation where we could conceivably have something like what was known as the Spanish flu from 100 years before, and it perhaps explains why there was such almost panic and sometimes some questionable decisions taken about how one could actually handle this epidemic. So it's something to think about. Okay, I want to look at some other diseases which occur in England and Wales, or have occurred in the 20th century, the first 90 years of the 20th century, and look at what's happened to them over time. And here we see 8 diseases, and it's quite possible that some of you have not even heard of some of these diseases, and if you have heard of them, you perhaps have never met anybody who's ever suffered from any of these, or at least some of them. And yet they were, some of them in the past, very, very serious problems. So something like diphtheria. I've never met anybody or even heard of anybody who has ever had diphtheria. And that's not terribly surprising because I was born in about 1946, which of course is a very long time ago. But even by that time, if you look at diphtheria, the top left-hand graph, you see that whereas it was actually really quite common in the first sort of 30, 40 years of the 20th century, it died off very, very quickly. And you might like to think about why that might have been. And the same sort of thing happens, but it was a major killer, diphtheria. I mean, when you got it, I mean, it's quite likely that you weren't just going to get slightly sick, you were going to die. It was really, really serious. Similarly with typhoid. Diphtheria has pretty much disappeared entirely. Typhoid still can hang around, but it's nowhere near as serious as it used to be. Measles are more interesting, and we'll talk about that a little bit later. And then we have polio, of which I'm sure you have heard, and you will still see people who suffered from polio. And you can see that basically it sort of hung around at a very low level between 1930 and 1940. In fact, pretty much until the end of the Second World War. And suddenly it went completely berserk. And then it disappears. And it disappears to pretty much zero by the 60s. In England and Wales, not necessarily everywhere else in the world. Scarlet fever dies off more slowly, but again, it drops down to very, very low levels, and whooping cough as well, and so on. And you might like, I mean, we're going to look at what actually, how things changed and what brought an end to these various diseases. But you might like to actually Google them and find out a little bit more about what was going on. Okay, so we can in fact look at them and find out what happened. And in fact, what you see is that it must be in the sometime in the 1930s, somebody actually developed a vaccine for diphtheria, and so it goes from between 40 and 70 cases per thousand in the 1930s down to absolutely zero by 1950, or very, very close to zero. So big sort of move there, and again, that is basically due to the development of a vaccine. For typhoid and paratyphoid, and you need to read about these things. I'm not going to go into what they're, but they're basically where this comes from. This is a virus, and it essentially it's transmitted via feces, and people basically getting fecal material on their fingers and getting it into their mouths, and that will cause a problem. And so that isn't sorted out by a vaccine or anything like that. It's basically by an improvement of sanitation and hygiene. So think back to dear old Semmelweis. Okay, sanitation is still incredibly important, not just for puerperal fever, but for things like typhoid and paratyphoid. Measles, much more interesting. So the number of cases, it sort of wanders around until 1950, and then there's a long period during which time it sort of fluctuates with a period of 2 years. Each high year was followed by a low year. The introduction of a vaccine in the 50s reduced the measles incidence greatly, but it didn't entirely eliminate the disease. Okay, and we're going to talk a little bit more about this. But what we do see is, again, we've got this business of here is a disease which has been really quite severely restricted and contained by virtue of the development of a vaccine. And this one is even more, sort of striking. So polio is a very interesting disease. I mean, it's an absolutely horrible disease, as you well know. It basically causes wasting of the muscles, and it can lead to, it was led to a very large number of deaths. But even in people who survived, they lost the use of limbs and so on, and it was a really horrible disease. The interesting thing is that between 1930 and towards the end of the Second World War, it hardly featured on the radar. I mean, it's sort of cases of 1 per 1,000, okay, which is very bad if you happen to be the one, but it's actually really quite a low rate. And then suddenly we see this big spike after the war, and going until the 60s, the middle 60s, when suddenly there is actually what was known as the Salk vaccine, and it absolutely knocked polio on the head. And right now it is virtually unknown in the United Kingdom, and there is a real possibility of actually eradicating polio entirely from the world, but it has not yet actually managed to be done. But just while we're thinking about this, one might like to there is, of course, a subsection of the population that thinks that vaccines are very sort of nasty and liable to cause all manner of abnormalities in children and so on and so forth, and people refuse to get their children vaccinated. If you look at something like polio and realize how appalling it was, you need to realize that the people in the anti-vaccine brigade need to think rather more carefully about what they're saying. There we are. But the interesting thing is that what actually happened was that there was what is thought is that curiously the massive increase in the 40s in the rates of polio were in fact as a consequence of increases in hygiene. Because what is said is that, of course, young babies tend to, sort of below the age of 3 or 4 are crawling around on the floor, and they're putting everything in their mouths, and they're putting all sorts of things in their mouths, and they're getting they're getting exposed to infections. And if in fact you get exposed to the polio germ when you're very young, what happens is you get a fever, but it doesn't last terribly long, and it doesn't cause any great problems. However, with the improvements in sanitation and hygiene, what happened was that there was much less chance of a baby contacting polio, or some of them did, but there was a good chance that other people who actually were living in sort of hygienic situations didn't actually contact the polio bug until they were rather older, and at that time it became very much more serious, and it was a real problem. So it's slightly counterintuitive, but it was taken care of very largely as a consequence of the vaccine. And penicillin, essentially, of course, what we need to know is that it wasn't simply vaccines which were developed in the 20th century. During the First World War, there would have been the part of the massive rates of death in the trenches was due to people who were getting these terrible injuries. But then, of course, they would be essentially dying because of infections, and there were no antibiotics. Of course, when penicillin was developed in the 20th century, this actually changed things really quite remarkably. So something like scarlet fever, for instance, there is actually a big reduction as a consequence of the use of penicillin. There are, by the way, if you go through these slides, you will see that there are notes which you can look at attached to a lot of these slides, which will give you more information than I'm giving you in this short lecture. Similar thing with whooping cough. Again, there is a vaccine developed, and where you see that written on the slide on the right hand side here, where it says panic, this is basically what happened was that there is a subgroup of the population, a lot in the United States in particular, who basically decided that vaccines were causing all manner of real problems in young children, and they simply refused to have their children vaccinated. And hence or otherwise, you had a rise in whooping cough, you had rises in measles and things like that. So it's a serious problem, but it's a much more difficult problem to talk about than I've got time to talk about at this time. So now we'll change things slightly. I mean, many of these diseases, as you saw, were somewhat eradicated by towards the end of the 20th century. But there are always other things which are lurking out there, and the massive pandemic which hit the world towards the end of the 20th century was, of course, the HIV AIDS epidemic. And this basically shows precisely how serious this can be in the number of deaths recorded in Baragwanath by age and sex and HIV status between the years 2006 2009. The number of the deaths are very huge, and you see that they show a very strong relationship with age. So you get very small numbers of deaths in the younger people, but it's because, of course, it's a sexually transmitted disease. Once people become sexually active in their late teens and into their 20s, then you see this massive spike in the numbers of deaths due to HIV. Okay. We're going to change gear now completely. You might be quite happy to know I'm not going to talk about disease anymore, but I'm still going to follow the same sort of theme that we've been talking about, and that is to sort of look at somebody who worked on a biological problem, looked at the data, formed a hypothesis, and came up with some ideas. And they turned out to be extremely important ideas. And the person we're going to talk about first is a monk who lived in Europe in what is now, I think, the Czech Republic, and he was interested in gardening, and he was interested in peas in particular. And he did some very interesting things with peas. What he discovered was uh, that you can find peas when you grow them, some of which have long stems and some of which have short stems, but they're never in between. It's either long or short. That seems to be it. And they breed true. That's to say that if you take tall plants and you cross them with other tall plants, and you grow them, you grow seeds from tall plants, then they always produce tall plants. And if you take the seeds from a short plant, that always produces a short plant. However, if we cross-pollinate these peas, a long, a tall stem, and we take that and pollinate it against a short stem, what happens when we cross them? And we get what's known as the first filial or the F1 generation. And then also what happens if we take these F1 plants and we cross them, and we get the F2 generation. And what he discovered was you only get two kinds of peas. You get tall or short. So if you take tall, as you look on the left-hand side, and you can grow them as many times as you like, and they always produce a tall plant. And you take a short plant and you pea, and they always produce another pea with a short stem. But now here's the interesting thing. If you take the pea with the tall stem and cross it with a pea with a short stem, then in fact all of the offspring are tall. Okay, you don't get some short and some tall. You don't get some, they're not in the middle. They're just all tall. On the other hand, if you take those ones and you actually then breed those ones to the F2, so if you cross these F1s together, you get an F2 generation, then in fact you get some which are tall and some which are short. Now this is very, very curious. And Mendel, he's doing much the same as Semmelweis, but in a rather more benign situation. And he's looking at this and saying, what on earth can possibly be going on here? And so he starts thinking about it. Okay. And what he does is he then looks not just at the length of stems, but he looks at seeds, and he finds out that the seeds come in two sorts of seeds. They're either round or they're wrinkled, and they're either yellow or they're green seeds, and they either are purple flowers or they're white flowers, and they're smooth or constricted pods and axial versus terminal flowers, etc., etc., etc. And what we saw in when we were looking at the business of the length of the stem was that if you crossed a purebred long-stemmed plant with a pea plant with a short-stemmed pea plant, you always got a long-stemmed pea. That is to say that we say that the long is dominant over the short. Okay, and when he looked at these various characters, round versus wrinkled, etc., etc., he found a lot of them where basically the dominance, you had a very strong dominance. And in fact, if you looked at the ratio of the dominance to the recessives, when you got the F2, you found that it was around about 3-to-1. Okay, so this is very interesting. So what he was basically finding, what did he say? Okay, well, we'll look at what he... So let's just think what it was that Mendel was saying. He's basically saying that you can, if you're talking about, for instance, the length of stem that you're going to get, you can get two variants of a gene, and they will call this variant alleles. So you can either get an allele for the tallness, or you can get which is the big T, or you can get one for a short stem, which is the small t. Okay. And if you cross two plants, both of which are tall and they're bred true for tall, so they've just basically just got big T from both sides. So you will get big T from both sides. So you will, if you breed peas which are both big T, what you will get is you'll get big T's, and if you use the small T's, you're going to get the short. So you're going to get longs and shorts, and they will breed true. If, however, you breed a plant which is basically bred true for the long stem, for capital T, against one which is breeding true for the short stem, so that's the two lowercase t's, then what you got is that basically from one plant you're always going to get a capital T, and from the other plant you're always going to get a lowercase t. So the offspring is going to be big T and little t, but because big T is dominant, all of the F1 generation are tall because T is dominant. But now look what happens when you actually take these plants, these F1s, which are all big T, little t, and you cross them. Now what happens is that you can get a big T from one plant and a big T from the other, or a small T from one plant and a small T from the other, or in fact you can get a big T from one and small t from the other, and vice versa. So you see in that slide what it looks like. So what happens is now, because T is dominant, what you will get is from out of the four that offspring that you get, 3 of them are going to look as if they are just entirely tall, and only one is going to look as if it's going to be a short plant. Okay. Now, the point was that although Mendel didn't really understand it, he had just actually discovered what we now know as genetics. He did at least write up the results of his experiments, and they were there for all to see. And in particular, they were there for one Charles Darwin to see over in England. Now, Mendel would have been writing in German, and Darwin, of course, would have been able to read English and maybe not so well the German. Nonetheless, it's pretty certain that was known that he had Mendel's paper, and he probably read it, but it didn't actually click with him that what Mendel had provided with him was a mechanism underlying his theory of evolution, which had worried him for all of his life. And this is very interesting. And so Charles Darwin has this rather, which we now see as being a strange view of how inheritance works. And he says the laws governing inheritance are for the most part unknown. No one can say why the same peculiarity in different individuals of the same species or in different species is sometimes inherited and sometimes not. Now you see, from his point of view, because Darwin was thinking that what you got when you had sexual reproduction is that you just got a mixing of what you got from the male and of the female, whereas what basically had come out of Gregor Mendel's experiments was that there is not a mixing. You get an allele from the male and one from the female, and they stay separate, and they are inherited separately into the F1 and F2 and the generation. They always stay separate, and it was because of that it made so difficult for Darwin to understand precisely what was actually going on. All it basically shows is that if you look really carefully at the biology, if you look closely enough at the biology, and you look at the data carefully enough, you can find the answer. But even for Darwin, from his side, looking at, even though he had access to Mendel's data, he couldn't actually see what was actually going on. So we should take courage that it is, even with the greats, sometimes they don't actually see things which are there to be seen. So this is an interesting thing, and hope you've enjoyed this little walk through some history of biological theory. So just to finish off, here is some advice to young epidemiologists, which has been taken from an article by Taylor and Wheeler in Space-Time Physics in 1963. And you might like to think about this and decide whether you actually think it's true or not true, or in each case. But what they say is that when you're doing modeling, don't make a calculation until you know the answer. Now that's quite a controversial thing to say, and you might like to think what they mean there. But what they're basically saying is make an estimate before every calculation, so you know what you think you should be getting, and try a simple biological argument. So, for instance, if you're trying to work out the R naught, the generation time, the selection, survival, control, and so on, and so forth. Guess the answer to every puzzle. And be courageous. You can guess, but you don't have to tell anybody. No one else needs to know what the guess is. But it's quite informative to make a guess and then do the calculation and then find out whether you're right or wrong. So you basically make a quick estimate, okay, just by instinct. And it's great if you make a right guess and it reinforces your instinct. On the other hand, a wrong guess makes you think again, and you think, well, that's really interesting. I wonder why that is. And you get back to look at things. And in either case, life as an epidemiologist is, however long, is lots of fun. And I think you'll discover that the longer that you work in this area. So enjoy. And just as a final summary of what we've looked at, we do emphasize yet again the importance of the data. It's all about data, data, data. Stay as close to the data as you can. And look for patterns. Look in there, there's going to be some interesting patterns. They were there for Gregor Mendel. And Darwin, even Darwin didn't see them. But you just keep looking at the data for the patterns. Look at them, at the data, and think about the data. And know that everything is going to be driven by the biology. It's all making complete sense. So you must put in as much biology as you can. If the more you know about the biology of the situation, the easier it will be for you to get an understanding about what is going on. And then, as we said, don't rush in and make massively complicated mathematical equations. Keep it simple. Do what Einstein tells you. Keep it simple. And always remember that the purpose of the modeling is not to fit the data, but to sharpen the question. Okay, well, thanks for your attention, and I hope you've enjoyed this. And just as a final word, I should say that whereas John Hargrove, actually made this presentation, the slides are entirely due to Brian Williams, with whom I've worked for many happy years, and many thanks to him, and thank you to you for listening. Okay.